So next is going to be Christophe. So Christophe comes from Marseille. He's in ex-Marseille, and basically he works on both seizure and also experimental setting in rodents. And what he's going to talk to us about today is about like multiple timescales and rhythmicity in body-brain interaction. So Christophe, please.
Thank you very much for the two questions which were shown on the first slide yesterday. And basically what I would like to cover today are some aspects of what kind of multimodal data is required for modeling the brain and how multimodal data should be structured. And my contribution to this is time. And basically what I would like to talk about are difficulties that we face when we study the brain. And I name here three among many, but three on which I'm working on right now. The first one is circadian rhythms. The second one is multidian rhythms, and the fact that the brain is not alone. So let's start with the circadian rhythms. As you all know, all living organisms on the planet that we know of show a circadian rhythmicity. Even cyanobacterium in the depth of the ocean, they have a circadian rhythmicity. So in humans, what it means is that many of our body functions are regulated in a circadian manner. For example, melatonin starts at 9, and that induces sleep. And for me, it's just right now, because it's close to 9 p.m. for me. So I'm going to fall asleep when I'm talking, so don't pay attention. And secretion of melatonin stops at 7:30. The high alertness is right now in the morning. Now for physical exercise, it's better in the afternoon and so on. All organs in the body follow circadian rhythmicity. Of course, brain is well known for that. The suprachiasmatic nucleus and Nobel Prize winner was about circadian rhythmicity described in the system. But all the other organs in the periphery, they have also circadian clocks. Even some are totally autonomous, like in the microbiota, as its own circadian clock, which means that many body activities are regulated in a circadian manner. They change from 1 hour to the next. Now, how this happens, we know that the suprachiasmatic nucleus is a key structure in the brain that regulates circadian rhythmicity, and it pilots, it controls the peripheral clocks outside the suprachiasmatic nucleus in the brain, but also that of peripheral organs. How does it happen in the suprachiasmatic nucleus? Basically, what has been demonstrated is that some clock genes are oscillating in a circadian manner from 1 hour to the next. They are up and down regulated, sometimes in phase opposition, like clock and PER2. And because they are transcription factors, many other genes have their expression regulated in a circadian manner. So the system is oscillating. The molecular architecture of the suprachiasmatic nucleus is changing from 1 hour to the next, and does that every day. So, of course, the question was, is it only the suprachiasmatic nucleus? And in these two papers in Science from the same group, they sacrificed mice every 4 hours, and they collected the forebrain, isolated synaptoneurosomes, mixing all brain regions. And what they found is that most of the transcripts in synaptoneurosomes are oscillating in a circadian manner. 2/3 of the proteins of synapses are oscillating during the night and day cycle. That's a lot. So from 1 hour to the next, your synapses don't have the same protein profile. Not only that, but also the phosphorylation of the proteins is also changing in the circadian manner. So no wonder that already 50 years ago, there was a science paper showing that synaptic transmission in vivo was changing in a circadian manner in the hippocampus. And we know that, for example, place cells, properties of a given place cell in the hippocampus is changing during the night and day cycle. So the properties of place field change in the circadian manner. So many functions and long-term potentiation also is regulated in the circadian manner, and learning and memory is regulated in the circadian manner. So time is essential to consider when we are studying brain function. When does it happen during the circadian cycle? And this is also important for epilepsy. So we got scooped by these two papers because we are doing the same thing, but in the hippocampus specifically, and we were comparing control and epilepsy. What we found is also that many genes, transcript, and proteins are oscillating in the circadian manner in control, but also in epilepsy, and it's a different set of genes which is oscillating. Now that poses a very complex issue for data interpretation. As you know, when we are studying neurological disorders or any disease in any organ, we tend to compare the control situation with the disease situation. And we do a difference. We look at the difference and we try to infer mechanisms. But if you have circadian cycles everywhere, like your proteins are changing from one hour to the next, how do you interpret data? So this is one example. This is the alpha-3 subunit of the GABA receptor. It's important for us in epilepsy. But if you look at how different it is between control and epilepsy, at 11 a.m., it's upregulated in epilepsy. But at 3 p.m., it's downregulated. So now, if these things are oscillating all the time, how do you compare two situations? At 11 a.m., you will find something. At 3 p.m., your system has changed, and it's now different, and you find different results. So this is important for the clinic because seizures are not random. So I'm going to show you the first scientific paper on epilepsy. It was published 5,000 years ago. It's a Babylonian tablet. And here, as you can read clearly, the doctor says that his patient had seizures always at the same time during the afternoon. It's interesting. Seizures are regulated in the circadian manner. By the way, in this scientific paper, they were publishing at that time the comments of the reviewers, which were here, and obviously the author didn't like them. So, in fact, 99% of the patients have a circadian regulation of the seizures. They always occur in a specific time frame during the night and day cycle. So this led me to propose a very stupid hypothesis, very simple. If the molecular architecture of your neural circuits is oscillating during the night and day cycle, then maybe at some time this network configuration is more seizure prone than at this time. So if you happen to have seizures in the afternoon, maybe your circuits in the afternoon are more excitable in some way. And that could be tested experimentally. That's what we're trying to do right now. So here, the response of the question, how should we structure? Well, we should consider time as an independent variable. And this is linked to what Jacob has shown in the question for him. If we want to model this, how can we model the dynamics of these proteins, which are regulated in a circadian manner, and how can we integrate that into our approach to understand how the system behaves? So, everybody knows about circadian rhythms, but I guess that few of you know about multidian rhythms. What are these things? Well, this has been discovered already a long time ago in trees, of course, because you have these annuli that are growing every year. But in bones and teeth and feathers, multidian rhythms have been identified. So this is a human tooth. It's difficult to see on this, but there are stripes of enamels which are deposited every day. So every day there is a new layer of enamel on the tooth. And every 7-8 days, there's a different enamel layer. And that's how, in anthropology and archaeology, anthropology, yes, they date fossils, okay? So these multidian rhythms are found also on feathers growth. It is believed that it's coming from metabolism, and the multidian rhythms are always a multiple of 24 hours. It depends on the size of the species. The multidian rhythm is in squirrel monkey 3 days up to 14 days in elephants, and humans, it's around 7 to 8 days. Okay, so you're going to tell me, well, that's modern, weak. Well, no, the multidian rhythms are found in Pliocene hominins 3 million years ago. And on feathers dinosaurs, you find evidence of multidian rhythm. So the circadian rhythm is there because the Earth is rotating in 24 hours. The multidian rhythm, we don't know where it's coming from, but it's there. And it's been there for millions and millions of years in species that we know of. Okay. Interestingly, now you can ask, why do we have a 1 week of 7 days? So maybe some ancestors had each 7 days layer of enamel that was making his teeth painful, and decided that he should rest this day, and that became the week of 7 days. But it fits with our inner clock. Maybe it's an inner clock of 7 to 8 days, and we just decided that we should have a week of 7 days. Otherwise, there's no reason why it should be 7 or 8 days. Okay, so this is the multidian rhythm for teeth, feathers, and bones. Why should we care? We are working on the brain. I mean, these scientists studying tooth, what they can teach us? Well, again, epilepsy was an entry point to this aspect, and quite recently, multidian rhythm were rediscovered. So these patients in the US are equipped with a stimulator, and this stimulator is also recording brain activity. That's for patients with intractable epilepsy. And there are hundreds and hundreds of patients equipped in the United States, and some have been recorded for decades. Now, the system is cool because not only it captures seizures and saves them, and you can retrieve the information, but at the same time, it's recording brain activity continuously. And in epilepsy, we have these pathological events that occur quite frequently that are called interictal spikes. They correspond to the firing of lots of neurons simultaneously. And they happen all the time. Seizures are rare events. Okay, and the system counts the number of spikes per hour. And this is what you see here. This is the number of spikes per hour. And this is one patient recording during 2 weeks. This is night and day, and you can see there's a circadian regulation of the number of spikes. A lot during the night and a few during the day. But if you look on a longer time period, you can see like an envelope. It goes up and then it goes down. Now let's zoom out of 2 years of recording of the same patient, and you can clearly see a multidian rhythm of 2 weeks. That is a clock during 2 years. Some patients recorded during 10 years, they have this rhythm that is fixed. Okay? Now, guess what? The most common multidian rhythm across patients is 7 days. And it's not related to the day of the week. So this multidian rhythm may be building upon this biological rhythm that we all have, this multidian rhythm that nobody knows about in terms of the mechanism. Now, the multidian rhythmicity is found across species, humans, of course, with different methods. It has been established with dogs with natural epilepsy, and we have shown it also in our animal models. This is important for the clinic because if you look at the circadian phase, so when seizures tend to occur during the day, the 24-hour cycle, and the multidian rhythm, the phase of the multidian rhythm, you can provide patients with a map of high seizure probability. So they know that every X days at this time period, they have very high risk to have seizures. Okay, so that's for the multidian rhythm. Nobody knows where it's coming from, it's there. But when we are studying an organism, like a mammal, we need to take that into account because if it's the same as the circadian rhythm, maybe the properties of the networks are changing on a very slow time scale. So another time scale to consider. Okay, and then the brain is not alone. And this is the work of Talon-Baudry, which was really inspiring for me. This is a very nice review from last year. And basically, the brain is receiving, as we know, signals from peripheral organs. And by the way, the vagus nerve, 80% of the fibers are ascending. So the periphery is sending signals to the brain. 20% only is descending. But it's bilateral communication. And what has been demonstrated, in particular here in humans, is that the activity of these peripheral organs, like the heart, breathing, and the gastric rhythm, because the stomach is generating a rhythm that is with the slow period, we find in the brain many regions which activity is directly modulated by the periphery. And for example, the alpha rhythm is phased on the stomach rhythm and not the other way around. So stomach rhythm plays a key role in controlling the alpha activity in the brain. So another thing that is interesting, they recorded patients with epilepsy with electrodes that allow recording spikes. And these are 3 neurons recorded in 3 thalamic regions. And 0 is the time of the heartbeat. And this is consecutive heartbeats, which are measured. And you can clearly see that this neuron is inhibited 200 milliseconds after each heartbeat. This neuron is excited, and this one as well. Now, let's forget about the heart. You record neurons in this region, and you find that a neuron is inhibited every second. So, wow, that's cool. We have here a kind of timekeeper or pacemaker. No, it's directly explained by the heartbeat. So if we don't have this information, we cannot interpret correctly what we are seeing at the single cell level. So this is what we started to do because I wanted to combine both things. We don't know where the multidian rhythm is coming from, and we don't know whether it happens in control animals, except in the teeth, but what about organs and the brain? So we designed a telemetry system that works in DC to record breathing, the heart, the stomach rhythm, the brain, core temperature, locomotion, and so on. Here I show an example of an epileptic rat, and this is the number of interactive spikes per hour. And it's long-term recordings, so 3 months. You can see the circadian rhythmicity, but also I don't have to show you statistics. You see the multidian rhythm. It's super clear. Now, if you look at the heart, this is the heart rhythm, very strong circadian rhythmicity that is known, but you can see by eye the multidian rhythm. You can see it in the breathing. You can see it in the gastric rhythm. I can't show you the statistics here for the time, but you can believe me that these organs also show multidian rhythmicity, not only in epilepsy, but also in control. So this is a wink to Scott, because we are also using these networks, and we fed all this data, and we can predict the next cycles of multidian activity and circadian, and we can even analyze what is happening during seizures, and seizures correspond to a line attractor, just for the fun. And now, something that is obtained by a student, which is unpublishable, because I thought, okay, if my animals, control animals, have multidian rhythmicity, can it explain some variability in the data when we do behavioral studies? So unfortunately, now the students do not accept to work on weekends, so the halls are weekends. But we did a very simple test, a cognitive test of spatial memory that can be done every day. The rat does not matter. It's not annoyed by repeating this test every day. Well, in fact, there are not enough cycles to do proper statistics, but you can see the multidian cycle. And we repeated that in 7 other animals, and they show the same rhythmicity. So cognitive performance seems to follow also a multidian cycle, which may explain the noise that we see when we do cognitive testing in mice and rats, because if they all have different multidian rhythms, they will be picked up at different times and have different effects. And just to finish, now we are combining these recordings with neuropixels, and we record at the same time the heart breathing, the stomach rhythm with lots of neurons. And I had a test for you. What do you observe? So I'll go to the end because that's the end of my time. These are the first neurons shown to be modulated by the stomach. So this in the subiculum, here in the dentate gyrus, this is one cycle of the gastric rhythm. You can see that these neurons are modulated by the gastric rhythm. So not only the heart and breathing, but the stomach also contribute to that. And another wink to Scott. We fed all the data to this same RLS. I never know how to say that. It's too complicated. But here we can show the phase flow, and we can identify the trajectories and the switch that is happening between REM sleep and slow wave sleep. And one of them is a line attractor, the other one is a point attractor, which tells you that one is super stable and one it can easily be kicked out. So to conclude, time at different time scale, I think that's very important to consider for the future. And we need a holistic approach to better interpret what we see in the brain. And we should not forget that we are dealing with rodents and maybe far away from what the human brain is doing.
All right, time for 2 questions while you're finding raising your hands. I have a question actually. Do you use this for prediction of seizure? Because if you have this sort of like a systematic change of excitability, for example, then you can even use a simple, I don't know, heart monitoring to predict what's going to happen in the seizure instead of having long-term monitoring of the brain.
Yes, in fact, we can predict not the seizure itself, but the high seizure risk. And we have an NIH grant with a friend in Madison, and we are killing the animals at the low risk period, high risk period, and we are going to do all the omics to see whether there are differences using epilepsy as its own control.
Okay. Well, hopefully NIH will still be there. All right. Any questions? Okay. Sachin, please just speak up.
So many children with epilepsy show also that kind of pattern. For absence seizures, nobody knows. Nobody has tested it. For this data to be acquired, you need patients with drug-resistant epilepsy. So the system that records the seizure is only for adults, for young adults, not kids. But now with wearable, we can use the technology to measure that kind of thing.
All right. Let's thank Christophe.